Hello students, this is Gina again with our last topic on how neurons work. So the last topic of neuron physiology is synapses, and the picture that you're looking at is a picture of many axon terminals synapsing on a target cell. This lecture refers to items in study question number 11 through number 13 and is contained in sections 7, 8, and 9 of chapter 11 in your textbook. So you may recall that we've already looked at 4 different topics of neuron physiology: the resting membrane potential, the local potential, the action potential, and the nerve impulse, or conducting the nerve impulse. Now we're going to look at synaptic transmission in a target cell. So let's first talk about what a synapse is. It's an intercellular junction between a neuron and any target cell. There are various types of target cells. A neuron could be a target cell, a muscle cell, or a glandular cell, a cell in a gland. And a neuron can act on these target cells either through a chemical synapse or an electrical synapse. And I see here that I already have a mistake. The chemical synapse is slower, as is my computer, than an electrical synapse. An electrical synapse is faster. So you may recall electrical and chemical signals. The electrical signals that they use we're going to talk about in a second, but you already know that there's a current that travels the length of a neuron, and that's through multiple action potentials occurring down the length of the axon and ending in the axon terminal. At the axon terminal, a chemical may be released called a neurotransmitter, so that will be a chemical signal. Another alternative is that at the axon terminal, ions are released, or I guess move, from the axon terminal into another target cell. And that happens through gap junctions, and I'm going to show you a picture of that. So let's first talk about synapses. The first cell that's sending the signal is called the presynaptic cell, and a nerve impulse on its length, and then it synapses on a target cell, but we call that target cell synaptic cell, and it is receiving the signal, either a chemical signal or an electrical signal. Now we can also look at of synapses, or the different types. The electrical synapse has both cells, the presynaptic and the postsynaptic cell, joined by junctions. So ions will flow through the gap junctions directly from a presynaptic neuron to a postsynaptic neuron. These ions, like sodium or possibly chlorine. This is quite fast, and it occurs in many parts of our brain, so in the central nervous system. Alternatively, the synapse between a presynaptic cell here and a postsynaptic is that there's a cleft located in between. So the cleft is a space, and a neurotransmitter or a chemical has to be released into the cleft from the presynaptic cell. Then it diffuses across the cleft and binds to a receptor on the postsynaptic neuron. This is a slower process because it takes time for the diffusion to occur, but it's very common, and there are advantages of electrical synapse and synapse, which I'll talk about. So first, let's look at the electrical synapse. The advantage of an electrical synapse, and the current can go. Normally, we would call this the axon terminal of the presynaptic neuron, and this is our postsynaptic neuron down here. If an impulse reaches this axon terminal, usually sodium comes in and stimulate another action potential, but there's no more membrane. So instead, what happens is the sodium goes into the postsynaptic cell and triggers an action potential synaptic cell. This is very quick. The diffusion of sodium across the gap junction is much faster than across a synaptic cleft, so that's the advantage. Now, it could be that the postsynaptic neuron an impulse in the opposite direction, but that's neither here nor there. It depends on what's happening. I mean, if it's in a motor or a sensory impulse, we're not going to worry about that too much. Now, the advantage is that there is no way for this signal to be altered once it reaches the postsynaptic cell. It's either sodium that's gone in, and so the postsynaptic cell is depolarized, or it's chlorine that goes, and then the cell is hyperpolarized, more negative. So there's no flexibility, no gradation, and I'm going to show you a chemical synapse and how there is some flexibility with those. So this is the chemical synapse, and I just want to look at the structure with you first. The drawing is slightly different than the other drawing. Here's the synaptic cell, the axon terminal. And the postsynaptic cell is bottom, and it's not really drawn all the way out here so that you can see all the membrane. The space in between is called the synaptic cleft. And the presynaptic cell is going to release neurotransmitter from secretory vesicles. We call them synaptic vesicle neuron, but it's still just a secretory vesicle. And the neurotransmitter is the cleft. The cleft, as I mentioned, is the space where the neurotransmitter has to diffuse across, and then the neurotransmitter binds directly on to a receptor that is specific for that neurotransmitter in the postsynaptic cell. Often these receptors are ion channels. They open when they bind to a neurotransmitter. We say that they are ligand gated or chemically gated. Could be ligand gated. Now there's some other structure I didn't mention, and these are the green channels in the, and we're going to look at what they do. Calcium has a gradient that's high on the outside and low on the inside, so it gets into the cell. I'll mention this one more time. Synapse works is that the presynaptic neuron is depolarized because an action potential reached the end of the neuron. So this depolarization will ultimately stimulate exocytosis and release of the neurotransmitter, as we mentioned. That's the little blue chemicals here. I'll circle one. I didn't do a very good job. I'll do it again. That's the neurotransmitter. There's a delay, 0.5 milliseconds. Now, when the neurotransmitters, these ion channels open, and sodium, which is just normally in the cleft, will diffuse down its concentration gradient. And so the sodium will diffuse to the postsynaptic cell, which is then depolarized. And if enough sodium gets in, then another action potential will be achieved. So that's only going to happen if it reaches positive volts. I'm sorry, negative 55 millivolts. So let's look at how a neurotransmitter can be released. Why does the synaptic vesicle actually release the neurotransmitter by exocytosis just because an action potential arrived there? Well, the reaction, presynaptic neuron, stimulates these calcium channels to open. So calcium into the cell. Now this triggers the synaptic vesicles to fuse the presynaptic membrane. And when they fuse the vesicles, the neurotransmitter is into the cleft. So that's how a chemical synapse works. And go through the steps one. See if you can come up with the answers for what these steps. So is showing depolarization of the presynaptic neuron. Relates the opening of this calcium channel. Calcium moves into the cell, causing the secretory to fuse and release the neurotransmitter. Number 5. Number 6 is the neurotransmitter diffusing across the cleft, and number 7 is the binding of the neurotransmitter on the receptor, which is located on the postsynaptic cell. So I broke, and I'm going to just move this for a second so you can see that in the slides, in the notes page, these are, all those steps are located. Does that make you dizzy when I move that around? I bet it does. Okay, let's talk about the advantages, types of, of chemical synapses. So, a disadvantage to a chemical synapse is that it's slower than electrical synapses. But there's so many advantages. First, the presynaptic neuron can vary how much neurotransmitter is released. That happens by a number of ways. One is maybe the neuron just makes more neurotransmitter. Alternatively, the more the presynaptic neuron is depolarized, or the more impulses that travel down the length, the more neurotransmitter will be released. So it's flexible and variable. The postsynaptic neuron can also make some changes. So the postsynaptic neuron can change its ability to respond to the emitter by changing how many receptors it has. Now, there are also different neurotransmitters throughout the body that are used by different neurons. Some neurotransmitters are excitatory and some are inhibitory, much like the way sodium is excitatory and chlorine is inhibitory. But whether or not a neurotransmitter is excitatory or inhibitory will reflect the neurotransmitter binds to. So we're going to look at different neurotransmitters and different types of synapses next. I'm going to introduce you to 3 different neurotransmitters. One is called acetylcholine, another is epinephrine, and the last one is gamma-aminobutyric acid, but we just call it GABA, so you don't have to worry about that. The name of a neuron and the synapse that it forms that releases acetylcholine is called a cholinergic synapse. When epinephrine is released, we call it an adrenergic or adrenergic neuron. And that's because adrenaline is a lot like epinephrine in terms of how it chemically acts. So they're related. Epinephrine, norepinephrine, and adrenaline we all refer to as an adrenergic neurotransmitter, neuron, and synapse. GABA is a GABA-ergic synapse. So when the neuron releases GABA, the synapse it makes is called GABA-ergic. It should be noted at this point that that all of the neurons in your body, each neuron I should say, can only release one type of neurotransmitter. So it either releases acetylcholine or adrenaline. It doesn't release both. However, a postsynaptic cell could receive input from many different types of neurons. So a postsynaptic cell could have lots of receptors for different neurotransmitters. Okay, let's talk about the actions of these neurotransmitters. Some actions are excitatory and some actions are inhibitory. When a neurotransmitter is excitatory, the neuron forms an excitatory synapse. The postsynaptic cell will be depolarized. 2 examples of this. Acetylcholine neurons or cholinergic neurons, when they synapse on skeletal muscle cells, the acetylcholine is always excitatory. Epinephrine or adrenaline is released from an adrenergic neuron to a cardiac muscle cell. The cardiac muscle cell is always depolarized or excited. So in order for our skeletal muscles to contract, they receive acetylcholine from a neuron. In order for your cardiac muscle cell to contract faster, actually, they receive epinephrine. Now there are some inhibitory synapses as well, and these inhibitory synapses different neurotransmitters. One is GABA. GABA is always an inhibitory neurotransmitter, and it will cause hyperpolarization on its target cell, meaning lower than negative 70 millivolts. Acetylcholine, on the other hand, we saw is already excitatory to skeletal muscle, but when a cholinergic neuron synapses on a heart muscle cell, that is same chemical, same neurotransmitter, acetylcholine is. So we want to look at why that is next. So we're going to go to that next. First, let's look at an inhibitory neurotransmitter. Okay, get out of the way. So an inhibitory GABA-ergic synapse means that this presynaptic, so it's got a positive charge on inside, and GABA is released. So it's released in the synaptic cleft. I should draw a little. So GABA travels across the synaptic cleft to the postsynaptic neuron. Now the receptor on the postsynaptic neuron is, and the ion that it is specific for is chlorine. Move from the outside of the cell to the cell concentration gradient. This is the major inhibitory neurons in our cortex are GABA-ergic, and they help suppress kind of our limbic system. And there's other things also that they work on. Okay, let's look at an excitatory synapse next. I got to get myself out of the way. Okay, so I'm going to use choline as an example for an excited neurotransmitter and synapse. We know that when a neuron releases acetylcholine to a skeletal muscle, that skeletal muscle cell is excited. Is it excited? Well, acetylcholine cell that has sodium channel receptors. So you've seen this kind of thing before. What happens is the acetylcholine, shown here again, diffuses across the cleft and it binds to open the channel and allowing sodium to enter the postsynaptic cell. Now the postsynaptic polarized inside, and if there's enough sodium that goes in, an action potential will happen. It has to reach threshold levels for that to happen. So that explains how acetylcholine is excitatory, but as I mentioned, acetylcholine can be inhibitory. So let's look at why that is and how that happens. When a cholinergic neuron synapses on a skeletal muscle cell, we know, sorry, we have another mistake. Of course we do. Okay, I fixed the mistake. Now it also cells to be. So an inhibitory response happens because in the case of a cardiac muscle cell, the receptor ion channel, that's what it is. That's the receptor. And so chlorine moves into the cardiac muscle cell, and the cardiac muscle that is more negative. It's less likely to become depolarized and less likely to fire an action potential. This is how your heart rate is slowed down, and the force of contraction of your heart is slowed down. There is a way that acetylcholine inhibitory. It's a little bit more complex. In this case, acetylcholine binds to a G protein linked receptor, which is shown right here. I'll put a big X on it. So acetylcholine binds, that initiates a second message inside, maybe cyclic AMP, maybe calcium, but the ultimate result of that is that enzymes necessary for contraction are controlled. And so cardiac muscle contraction is decreased. So I'm going to say that one more time. Acetylcholine could bind to a G protein linked receptor. This receptor acts, when it's bound to the neurotransmitter, acts to decrease cardiac muscle contraction because it can control the enzymes that are necessary for. I really am more interested in you that neurotransmitter can be inhibitory when it opens the channel. Now let's look at a neurotransmitter or chemical synaptic transmission. Stop. How does it stop? It stops because the neuron diffuses out of the synapse, shown by number 1 over here. Maybe it's degraded. In some cases, it is taken into the pre-cell. So it's here to be reused. So when it diffuses out of the cleft, it eventually will be degraded. When it's degraded right in the cleft, it means that there's an enzyme in the cleft that does just that. An example is an acetylcholinesterase, which breaks down acetylcholine. Now when the neurotransmitter up into the presynaptic neuron, means the presynaptic neuron takes up the neurotransmitter. And for this to happen, there's usually a transporter protein that's required. Either the transporter is in the membrane here, or neurotransmitter transporter that's present, example of one of those. So you may have heard of a neurotransmitter called serotonin or dopamine. Maybe you've heard of those neurotransmitters. Those neurotransmitters both have reuptake transporters that are present in the synaptic cleft. So, for example, this red circle here, this red circle, I can't seem to circle it. Try again. It's not working. Okay, the neurotransmitter has released from the presynaptic neuron, and it binds to a receptor that's on the postsynaptic cell. Sorry, I had technical difficulties again. So the presynaptic releases the neurotransmitter, and the neurotransmitter there we go. The neurotransmitter is released to the synaptic cleft, and normally it's going to bind to a receptor that's on the postsynaptic cell, which I kind of just outlined. Whatever effect, excitatory or inhibitory, it depends on the receptor and the neurotransmitter. But a certain amount of the neurotransmitter that's been released will bind to these transport proteins that take the neurotransmitter back up into the presynaptic cell. This happens with serotonin and it happens with dopamine. I'm sure the ones I know about. Let's look at something called selective serotonin reuptake inhibitors. So these are called SSRIs in the psychiatric world, and you may have heard of SSRIs before. The point in taking a selective serotonin reuptake inhibitor, a medication, is that then serotonin would remain in the synaptic cleft longer. This will increase the response of the postsynaptic cell. So this is often used in the treatment of depression. It can also be used in the treatment of Parkinson's disease. There's a number of things that it can be used for, these SSRIs, or at least reuptake inhibitors. So what they do is they block reabsorption. Usually they act by binding to the transporter. So there's competition and the neurotransmitter can't get back in. If this neurotransmitter stays in the cleft longer because it's not taken back up into the presynaptic, then this postsynaptic cell will fire more okay, or it'll receive more ions and more likely be excited. And if this is in a pleasure pathway or a motor path, the responses are good, right? You'll move easier, you'll feel better. So that's the idea behind selective serotonin reuptake inhibitors. Now there is an example where this is a problem, and that is in a habituation. Talk about habituation a little bit. When neurons need to respond to it. So normally they're only exposed for a brief time, they respond, and it's over. If they are exposed to a neurotransmitter or repeated firing of that presynaptic neuron occurs, they are inundated with neurotransmitter. Now, due to this prolonged stimulation, membrane, it's known as downregulated. So the idea, I guess you can think of it as that the neuron's tired, okay, and it knows, I don't know if it knows, but it decreases how much stimulation just by not responding to what's out there. It's kind of like when I turn my cell phone off, okay? In this case, the postsynaptic cell is decreasing how well it can receive a signal by decreasing the number of receptors. And there are two conditions under which this has some sort of clinical relevance that I think you might be interested in. One is with prolonged use of SSRIs, and the other is with cocaine. Prolonged SSI use can cause that I'm going to show you with cocaine use, but it's a correct the problem when it's mentioned, because normally what will happen, we'll just stop giving the patient that particular SSRI and give them something else. And there won't be this terrible, I mean, there's a little bit of a transition period, but it's not a terrible withdrawal or anything. And it's all for health, right? But with cocaine use, this is a problem. So it makes addiction very difficult to overcome. Okay, so let's look at what happens in addiction. So in a normal synapse, let's say, I'm going to look at this first box here. You have a certain amount of neurotransmitter. I count 6 molecules. And you have 4 receptors. And due to the fact that the transporter is there, 1 in 4 receptors are stimulated, and then you get an appropriate response. Okay, so an appropriate excitation. Now let's say instead we're going to go over to this second box. Okay, let's say that somebody uses cocaine early on. I mean, they're early in their use. What happens is you still have 6 molecules of neurotransmitter, and this is showing maybe 5 instead of 4, but I guess on the previous picture there was another one stuck over there. But nonetheless, what happens is that cocaine binds to the transporter. That makes this neurotransmitter stay in the cleft longer and therefore activate more receptors, twice as much, 3 times as much. So you get an enhanced, I'm never going to be able to draw that, an enhanced response. So excitation is increased. So that's what happens with initial cocaine use. And if this is in a pleasure pathway and the neuron is excited overly, then you feel great, right? And with prolonged cocaine use, you may have heard, I'm going to go down to this third slide, that what happens is the amount of cocaine somebody needs to feel that good again increases. They sort of become resistant, right? Well, what happens in this case is that the postsynaptic cell has been so stimulated that it downregulates the number of receptors. Meanwhile, the presynaptic neuron is releasing the same amount of neurotransmitter as happened all the way through. But there's less of a chance for a response in the postsynaptic cell because the cocaine is there, but less than the second box, but more than the first box. So they still feel better than they did prior to ever taking cocaine, but they don't feel as good as they did early on. So that ups how much they use. What's even worse is that over time, if you take away the cocaine, so I'm in the fourth box now, somebody withdraws from cocaine, they want to stop taking it altogether. Now the transporters are working normally. The presynaptic cell has released approximately the same amount of neurotransmitter, maybe a little bit more, but basically the same amount. But the transporters are moving the neurotransmitter back into the presynaptic neuron. That means you have less neurotransmitter in the cleft for a less amount of time. And it's less likely that you're going to get binding to a receptor because the number of receptors has decreased so drastically. So if I take off all these silly markings, a normal with a cocaine withdrawal, that here you've got about a 25, these numbers up, but you've got about a 25% chance that this postsynaptic cell will be excited because the transporter is 4 receptors, certain amount of neurotransmitter. In the case with cocaine withdrawal, because the transmitters are transporters, there's a much less receptors. They've been downregulated. So everything that I just talked about is written on the next slide and your last slide. I'm going to show it to you. The addiction and not being able to withdraw is because and it's all due to the fact that the number of receptors decrease due to downregulation because dopamine or the neurotransmitter has been left in the because the so that's the end of this particular lecture. Thank you very much.